Hello and welcome. My name is Nathan Layle. I'm a scientist in Cayman Chemical's Forensic Chemistry Division. Today we're going to talk about the results of our human liver microsome studies to identify metabolites of semi-synthetic cannabinoids Δ10-THC and Δ6a 10a THC. Since not much is known about the metabolism of these semi-synthetic cannabinoids, we wanted to expose them to human liver microsomes to see what metabolites formed. The Δ9-THC structure here is shown to include the numbering just so you can understand how the numbering system works for semi-synthetic cannabinoids. And the cannabinoids that were involved in this study are shown on the right. So we included the two diastereomers, the 6AR9R Δ10 and the 6AR9S Δ10 THCs, as well as the two enantiomers, the 9R and the 9S Δ6a 10a THC. Our in vitro incubations were run using mixed pooled human liver microsomes. Incubations were run for 60 minutes at 37°C. The reactions were quenched with acetonitrile and then spun down and injected on an Orbitrap to help identify what those phase 1 metabolites are. The reason that we care about these semi-synthetic cannabinoids is because they continue to emerge on the recreational market. So the products shown on the right are some examples of products that are being marketed to contain specifically Δ10-THC or Δ6a 10a THC. The literature data really shows that the Δ10-THC diastereomers lack significant psychoactivity, but we know that Δ10-THC can isomerize further to give us the Δ6a 10a. And the literature does show that the 9S Δ6a 10a has about 10 to 30% of the psychoactivity of Δ9-THC. Recent metabolism studies on these types of semi-synthetic cannabinoids have been more focused on the detection and the quantification of carboxy Δ10-THC. And so our study hopes to inform us more of what hydroxylated metabolites form using human liver microsomes. Since these semi-synthetic cannabinoids have emerged so recently on the recreational market, our understanding of their human metabolism is limited and based solely on our understanding of the way Δ9-THC is metabolized. We know that the human liver contains a variety of CYP isoforms, and research on Δ9 indicates that CYP2C9 and 2C19 are responsible for the major oxidative metabolism of Δ9 to give 11-hydroxy-Δ9-THC. Isoform 3A4 is responsible for oxidizing Δ9-THC to the minor metabolite 8-hydroxy-Δ9-THC. There are other enzymes involved in the conversion of 11-hydroxy-Δ9-THC to the 11-nor-9-carboxy-Δ9-THC, and even less is known about the enzymes involved in converting Δ9-THC to a tail chain hydroxylated metabolite. We included in our study the masses for the dihydroxylated and the carboxylated species, but with limited evidence of their formation, we decided to focus solely on the monohydroxylated metabolites. So let's get into what our human liver microsome studies actually showed us. We're going to look first at the Δ10-THC diastereomers, starting with the 6AR9R Δ10-THC metabolites. Our chromatogram showed us that two major and two minor peaks formed, and we're going to focus first on the retention time 13.86 minute peak. The mass spec data shows that we have likely formed the 11th position hydroxylated metabolite based on the formation of ions 95, 109, 301, and specifically 193, which is the base peak of our reference standard, the 6AR9R 11-hydroxy-Δ10 standard. We also have consistent retention times between the standard at 13.84 minutes and the newly formed metabolite at 13.86 minutes. There is some ion ratio difference between the reference standard mass spec and that of the data we collected, which indicates that there's possible coelution with other hydroxylated species in this peak. When we look at our next most abundant peak, the peak eluting at 14.16 minutes, we see a mass spectrum that indicates that we have hydroxylation elsewhere on the ring system. So the retention time we know is different from our reference standard at 13.84 minutes. And so we suspect hydroxylation at either the 7 or the 8 position. We also see a very high abundance of 331 relative to 313. The 331 is the molecular ion, and the 313 is the water elimination ion when we lose the hydroxyl group. And what it shows, based on our understanding of how Δ9-THC fragments, is that our hydroxylation has occurred further away from the double bond, whereas in Δ9-THC, we see a base peak of 313 because the allylic hydroxyl group can eliminate rapidly. When we look at the minor metabolites that formed, we can see that in the case of the earliest eluting peak at 13.35 minutes, we seem to have formed the tail chain hydroxylated metabolite. And this is evidenced by the mass spectrum showing a key 191 fragment. So the drug testing and analysis reference that's included here is research done on hexahydrocannabinols, but they also identified that when you get formation of a tail chain hydroxylated metabolite, that you get elimination of that water to form the unsaturated ion, giving you the 191 peak. We also see that here. The other minor peak at 13.7 minutes is just consistent with another ring hydroxylated metabolite. We aren't able to establish really where that is, but we see some of these key ions like the 331 molecular ion, as well as the 193 and the 109 ions. When we look at the metabolism of the other Δ10 diastereomer, the 6AR9S Δ10, we observe the formation of one major and two minor peaks. The earliest eluting minor peak at 13.21 minutes has a mass spectrum that indicates we're getting tail chain hydroxylation. So we see that 191 and the 257 ions, which are diagnostic of the tail chain hydroxylated metabolite. Our spectrum also shows a minor 193 ion, which suggests we could be getting coelution of both tail chain and ring hydroxylated species. There's a lot of research that's been done on the metabolism of various cannabinoids, and this particular Journal of Natural Products study on CBC identified benzylic hydroxylation at the 1 double prime position. They were able to confirm this with their own synthesized reference standard, but other research in these areas indicates hydroxylation further down the tail chain in the 4' and the 5' positions. It's not clear where our hydroxylation is occurring, but our mass spectrum show that we are getting the tail chain hydroxylated species based on those 191 and 257 ions. When we look at the next most abundant peak, the peak at 13.57 minutes, we see in our mass spectrum the 193 ion, which indicates hydroxylation on the ring system. Now the peak shape and the high abundance of the molecular ion 331 indicates we could have a mixture of hydroxylated species. In this case, it could be the 11 hydroxylated metabolite or the 7 or 8 position hydroxylated metabolite. It's not quite clear which metabolite we have, and without a reference standard we can't confirm, but we can infer that with the high abundance of 313 relative to the 331 ion, we may have hydroxylation in closer proximity to that Δ10 double bond, and we may actually be seeing the formation of the 11-hydroxy Δ10-THC metabolite. When we look at the last most abundant peak at 14.13 minutes, we again have the evidence to support the formation of a ring hydroxylated product. We see a very strong 193 ion, nearly the base peak, alongside a very abundant 331 molecular ion. So we could be getting the 7 or 8 position hydroxylation, but it's also possible that we have 11 hydroxylation here. Now this spectrum is actually far more consistent with the 9R 11 hydroxy Δ10, and so this could be further evidence that that's the metabolite that we're forming. When we look at the metabolism of the 9R and the 9S Δ6a 10-THC, we see two chromatograms that are very similar. The 9R and the 9S are enantiomers of each other, and without any chiral separation techniques, we would expect them to behave very similarly. Our chromatograms show the formation of one major and one minor peak. These compounds have consistent retention times, and the mass spectrum are very similar for both of these compounds, with only slight differences in the peak heights between the 9R and the 9S enantiomers. The earliest eluting peak in both cases, at 13.31 minutes, has the key ions that we look for when we see tail chain hydroxylation, the 191 and the 257 ion. When we look at the major metabolite peak at 13.7 minutes, we have indications of ring hydroxylation. Once again, we have a base peak 193 ion, which is more consistent with what we saw for the 9R 11 hydroxy Δ10. So the mass spectrum and the ion abundances are very consistent with that and could be indicative that we are forming that 11-hydroxy metabolite, which is sort of what we expect based on what we know about Δ9-THC and its metabolism. And you can see that the 11-hydroxy Δ10 mass spectrum is here with a base peak 193 and sort of this staircase of the 301, the 313, and the 331 ions. Ultimately, our human liver microsome metabolism study on these semi-synthetic cannabinoids helped us to identify metabolites that we expect to find based on our understanding of Δ9-THC and the way it metabolizes. We found evidence of hydroxylation at the 11 position, on the tail chain, and elsewhere around the cyclohexene ring. Now, if you're looking to identify some of these key metabolites, you would look for, in the cases of tail chain hydroxylation, that key 191 ion as well as the 257 ion. We also noted some of these differences between the elimination mass 313 and the molecular ion 331. So if you have a very high 313 ion abundance, you would expect your hydroxylation to have occurred a little closer to the double bond. Whereas if you have a high 331 ion abundance and you have a much smaller 313 ion, seems to be indicative of hydroxylation further away from that double bond. And again, we know based on reference standards and literature that 11-hydroxy Δ9-THC has a base peak of 313 because of that allylic elimination, whereas 11-hydroxy Δ10 has a base peak of 193. And so these are the key ions you would look out for if you're analyzing samples that may contain semi-synthetic cannabinoids. These human liver microsome studies were conducted with the help and expertise of scientists in Cayman Chemical's bioanalytical, ISO-QC, and forensic chemistry departments. So we all appreciate your time and attention, and we hope you've learned something about the in vitro metabolism of semi-synthetic cannabinoids.